Assalamu alaikum, dear students. Hoping you will be enjoying your vacations, Corona as well as the winter vacations. Uh, Today, I will go through the lecture of lecture regarding carbohydrates. Carbohydrates are we need to look on its structure and the biological functions as well as its chemical reactions. You people know the glucose, fructose, sucrose, starch, cellulose, etc. These are the common words in our everyday life use, and they are the most abundant members of the large and important class of the biomolecules. These biomolecules are known as the carbohydrates. Um, Carbohydrates are defined as the compounds having the polyhydroxy aldehyde or the ketones. They contain the polyhydroxy means many hydroxyl groups in their structures, as well as they may be the aldehydes are the ketones molecules. The hydroxyl group provide the possibility of the strong intra and inter chain interactions through the hydrogen bonds. Through the hydrogen bonds. Just one minute. Just because of the hydrogen bonds. In addition to predominant elements carbon, hydrogen, and oxygen, some other molecules like sulfur, nitrogen, and phosphorus may also be present in these molecular structures. Later on, I will explain how this structure can be present. Like the other major classes of the biomolecules, proteins, nucleic acids, lipids, the carbohydrates are found in all forms of the life and serve many functions, many different functions. Like proteins, some carbohydrates perform structural functions, and some may perform the storage function. Under the storage carbohydrates, glycogen, starch, and are the important. While the structural function containing carbohydrates may be involved in making the DNA, making the protein, in making the RNA, in making the glycoproteins, in making the glycolipids, etc. There are some diseases may be associated with the carbohydrates, like the diabetes mellitus, which may be because of the uh, storage issues of the carbohydrates. Galactosemia, inability to metabolize the galactose, inherited defect in the enzymes. Glycogen storage disease, like a group of inherited, this is a group of inherited diseases characterized by the deficient mobilization of the glycogen. Glycogen may deposit on the different parts of the body in abnormal forms, and glycogen may lead to the muscular uh, and this abnormal storage deposition of the glycogen may lead to the muscle weakness and eventually and even to the death. Lactose intolerance, lactase deficiency, and this may be because of the lactase deficiency uh, and may lead to the diarrhea and intestinal discomfort. This is because of the deficiency of the lactase enzyme, which is important to digest the milk, which is important to break down the lactose into monosaccharides. On the basis of the units attached, carbohydrates may be divided into the monosaccharides, polysaccharides, and oligosaccharides. The simplest carbohydrates, sometimes referred to as the monosaccharides, are sugars, and either they are polyhydroxyaldehydes or the polyhydroxyketones. They can be derived from polyalcohols by the oxidation of one carbonyl and carbonyl group. For example, if you can see simple 3-carbon triol glycerol, let's say for example, the molecule can be converted either to the aldotriose or glyceraldehyde or to the ketotriose. The dihydroxyacetones by losing of the two hydrogen molecules, by losing of the two hydrogen atoms. So for example, this is you can see this is the glycogen. This is the glycerol molecule, which is an alcohol. By losing the two hydrogen, it can be converted into the into the dihydroxyacetone, which is the ketotriose, maybe because of the release of the two hydrogen atoms into the glyceraldehyde, which is the aldotriose. Since the middle carbon of the glyceraldehyde is connected to the four different substituent, because here you can see this is the middle carbon. This is the middle carbon, and this is attached to the carbonyl. This is attached to the uh, you can see the ketone. You can see the aldehyde group, carbonyl group. You can say it. Okay. Um, mm, Here, the central carbon is attached with the four different molecules. With the four different molecules, on one side it is attached to the alcoholic carbon, and on other side it is attached to the carbonyl carbon, and in this side it is attached with the hydroxyl group, and in this side it is attached with the hydrogen group. So this carbon is actually making the center of this molecule, making the center of this molecule. So this is the and attached with the four different groups, four different carbons, because this carbon is attached with the four different carbons. So this is making the chiral center. So this is the chiral center. It is which may lead this chiral center may lead it into the two different forms of the carbon, which may be called the stereoisomers, or you can call it the enantiomers. It may be present in the D form or the L form. You can call it the dextrorotatory or the levorotatory form. We may visualize the 4, 5, or 6 carbon sugars as arising from the triose through the stepwise condensation of the formaldehyde to the either glyceraldehyde or the dihydroxyacetone. From the smaller molecules by joining the other molecules through the reaction of the aldehyde with the alcoholic group, with the alcoholic group. From the smaller molecules, larger molecules can be made. So condensation reactions takes place in this process. So, for example, here is the glyceraldehyde. Through the process of the condensation with the one carbon molecule, it may be converted into the four carbon compound by the condensation with the some other molecule, and getting one carbon from that particular molecule. It can be converted into four carbon, which may be the erythrose or the three halos. So from this erythrose with the condensation with one carbon may convert into the five carbon compound, and similarly for this three years it may also be converted into the five carbon compound, which may be the ribose, rabinose in case of the erythrose, and xylulose and the lactose in the case of the threose. If one more carbon is added, is condensed with this molecule, it can be converted. Arabinose can be converted into the glucose and mannose, and ribose can be converted into the allose and the antrose. So by xylulose again we can get the we can get the glucose, iodose, galactose, and tallose can be obtained from the lactose molecule after the conversion. So here you can see from the aldehyde compound we are getting this one erythrose, and this is the three arrows. From the erythrose we are getting the CHO, ribose, and the arabinose, and from this we are getting the allose and aldose molecules. Monosaccharides are cyclized to form the hemiacetal. Aldehydes can add hydroxyl compound to the carbonyl compound. By this addition, the closure of the chain may take place. Uh, if a molecule of water is added, the product is an aldehyde hydrate, and if a molecule of alcohol is added, the product is the hemiacetal. Addition of water may hydrate the molecule, but addition of alcohol may make the hemiacetal molecule. Further, the addition of second alcohol result in the acetate. Okay, you can see this is the okay. This one is the aldehyde molecule. Aldehyde molecule. This one is the aldehyde molecule. If we add the water in this molecule, This molecule may be hydrated and it will make the aldehyde hydrate. But if we add the alcohol in this molecule, this alcohol will make it hemiacetal. This alcohol will convert it into the hemiacetal. While the further addition of the alcohol, it will convert it to the acetal. Acetal it means that hemiacetal mean at one position the acetyl group will be added, and the complete acetal mean on the both the hydroxyl groups on the both of the position one position of the hydroxyl group and on the other position where this double bond oxygen was present, it will be converted into the an alkyl group will also be added over there. Same is the case for the hemiketal group. If the alcohol by react, if the keto sugar by reacting with the alcohol is converted by adding if the keto group is um, by addition of the alcohol is converted or contain or obtain the one alkyl group, it may call the hemiketal group. The carbonyl group of the ketone react with an hydroxyl group in the similar fashion to form a hemiketal group. Okay, here we see it will form the hemiketal group. Okay. Okay. Now, we move further on the structure of the glucose. We'll discuss later about the hemiketal and hemiacetal form later on. The structure of the glucose uh, is explained mainly by the three scientists known as the Emil Fischer, Levin Hayward, and the Frudenberg. Emil Fischer, explained the structure of the glucose, and he got the Nobel Prize in the chemistry for the explanation of monosaccharide structure. Uh, the structure of the Fischer is the best used to represent the state chain structure of the monosaccharides, and its 3D diagram, the molecule diagram, as a flat structure is very important. Um, okay. He explained the configuration of the asymmetric carbon atoms in the aldehyde doses, and on the basis of its projections, roll and stick model was made for the explanation of the glucose structure. According to the Fischer, carbohydrate CHO molecules, carbonyl molecule CHO, you can call the aldehyde compound, aldehyde molecule, aldehyde carbon, and the alcoholic carbon are extended away from the holder, while the other four carbons are arranged in a line in between the alcoholic and the carbonyl carbon. The remaining molecules are the hydrogen and the hydroxyl groups. These hydrogen and the hydroxyl group are extended away from the other. If say for example, if we have a paper plane, and we extend these molecule on this plane, and we extend this on this plane on this plane. Say for example, here will be the carbonyl molecule, carbonyl carbon, and here will be the alcoholic carbon. Here will be the alcoholic carbon. Remaining four carbon molecules will be distributed between the carbonyl carbon and the alcoholic carbon. Remaining are the bulky groups, which are the H and the hydroxyl group. Both of these compounds are protruding away from the holder. They are present either on this side, either on this side of the molecule. Okay, this is a structure of the molecule. Yeah, explain later. Glucose has the two crystalline forms. The crystals form the water, are known as the form is known as the alpha form, while the crystals made with the acetic acid make the beta form of the glucose. The difference is among their rotation. The alpha form contains the 113 degree rotation up to some extent 112.9 or 113 rounding of the data. Okay, rotation of the beta form is the plus 19 degree. After sometimes while standing together, they may interconvert into each other, and the solution may contain the 52.5 and degree sugar. Okay, the solution may contain some molecules at the 52.5 degree. This procedure of interconversion of the alpha into the beta form is known as the Noether rotation. But through the Fisher's projection, the these two anomeric forms of the sugar glucose cannot be explained. These two anomeric forms of the glucose were explained by the Hayward formula. According to the Hayward, how these two forms can be obtained. I was telling you about the hemiacetal and hemiketal form of the molecules. Uh, Hayward explained the hemiacetal and the hemiketal form of the molecule. According to the Hayward, aldohexoses and the other sugars react internally and make their cyclic structure. Say, for example, the inner carbonyl carbon of the glucose molecule and the inner alcoholic carbon of the glucose molecule will react with each other and make the hemiacetal form. Will react with each other and make the hemiacetal form, leading towards the closure of the molecule. And it may close the molecule to make the ring structure. According to the Hayward formula, bonding of the fourth and the fifth carbon move the fifth carbon group at a position where it can react with the aldehyde group of the carbon number one. Okay, say for example, if we draw the structure, uh, say for example, this one here, and this one. This say for example, this is carbon number one, this is carbon number two, this is carbon number three, this is carbon number four, this is carbon number five, and here is the carbon number six. Uh, Reaction of the carbon number four and carbon number five move the carbon number five on a place where its hydroxyl group reached at the where its hydroxyl group may be at the position of maybe near to the carbon number one. May be near to the carbon number one. Here is the carbon number one. Maybe near to the carbon number one. Okay, because this hydroxyl group is close to the is close to this carbon, this hydroxyl group will react with this carbon. Reaction will take place between these two. Because of this reaction, this open ring, this open chain will be closed. Because of this reaction, this open chain will be closed, uh, and a structure will be made. This open chain will be closed. Okay. While the six carbon compound while the six carbon compound is present above this plane, the six carbon is present above this plane, above the carbon number five. Okay. So bonding of the fourth and the fifth carbon move the fifth carbon hydroxyl group at the position where it can react with the aldehyde group of the carbon number one. Here is the carbon number one. Here is the carbon number one, where it can react with the hydroxyl group of the carbon number five. When the reaction between this hydroxyl group and this aldehyde carbon will take place, the ring will close. Ring will be closed. Okay, this is a closure of ring. Okay. As hemiacetal formed, the two stereomeric compounds alpha and beta can be differentiated. Okay, after the closure of this chain, after the closure of this chain, and the cyclic when the cyclic structure is formed, now we can differentiate between the alpha and the beta form of the carbon, beta form of the glucose molecule. According to the Hayward projections, if the hydroxyl group is lying on the lower side of the structure, this will be the alpha form. It will be the alpha form. And if this hydroxyl group is present on the upper side of the structure. On the upper side of the structure, this will be the beta form of the molecule. While generally, to explain the general structure, we can only write a hydroxyl group in the form. But if we need to explain the structure, if we need to differentiate the structure for the hydroxyl group, for the beta, we will write the hydroxyl group on the upper side. For the alpha, we will write the hydroxyl group on the lower side. Freudenberg proposed alpha and beta on the basis of configuration of the relation of the anomeric hydroxyl group to the reference carbon. He explained that alpha anomer, if the anomeric hydroxyl group will be on the same side, it will be on the same side of the reference carbon. If the anomeric hydroxyl group, if the anomeric hydroxyl group, say for example this one is the hydroxyl group, will be on the same side of the anomeric hydroxyl group, will be on the same side of the anomeric hydroxyl group, the structure will be the alpha. If it's vice versa, if these are the opposite, if the alpha anomeric is on the other side, then the reference carbon, the structure will be beta. The structure will be the beta. This structure will be the beta anomer. How we will differentiate with the anomeric carbon? The anomeric carbon is the carbon which may be attached with the two oxygen atoms, which may be attached with the two oxygen atoms is called the anomeric carbon. While the reference carbon is the last chiral carbon. The reference carbon is the last chiral carbon. These are the definitions of the two molecules, two carbon compound. Anomeric carbon which may be attached to the two oxygens and which may be attached to the two oxygens, and the last chiral carbon is the reference carbon. So this one is the last. So for example, then this one is the last chiral carbon, and uh, this one is the carbon which is attached to the two oxygen. One oxygen of this hydroxyl group and second oxygen is this one. So this is the anomeric carbon. Okay. And the last carbon is known as the reference carbon. If the hydroxyl group of both of these are on the same side, if the hydroxyl group of both of these are on the same side, then this is known as if the hydroxyl group of both of these are on the same side, then the anomer will be the alpha anomer, and if the hydroxyl group of both of these will be opposite sides, one on this side and second one on this side, uh, it will be beta anomer. Final aspect, final aspects. Of the glucose structure are there are two final aspects of the glucose structure. One is the chair and second one is the board form. In the chair structure, triple one degree rotation, triple one bond between the carbon oxygen and carbon are found. In the chair structure, the minimize internal stress. Chair structure minimize the internal stress. Bulky groups are present equatorially, so the structure is stable. While the boat structure is not stable structure, it contains 109 degree between the molecule between the carbon atoms. Bulky groups are rotated present upward of the molecule and close to each other because of the van der Waals forces. They are too close and to each other that there may be an internal stress can be present, so the structure is unfavorable. This is the chair structure of the molecule. This is the chair structure, and this one is the board structure. This one is the board structure. Okay, here we can see the carbon molecules are present equatorially. while in the board structure, the axial presence of the molecule can be noted, while the bulky groups are present upwards. Here you can see bulky groups are present upwards. So, making the upward, and then because of their presence, because of this structure, there may be a van der Waal interaction between these molecules, and because of this interaction, the internal stress is created on the molecule, so the structure is energetically unfavorable. Reactions of the monosaccharides. So monosaccharides can perform many different reactions. Among this reaction, the major reactions are oxidation and the reduction reaction, the complete metabolic breakdown of the glucose to the carbohydrates and and the water is a process involving the several oxidation steps. Mm, the detail of which can be will be discussed later on. The functional group most susceptible to the oxidation is the aldehyde group, which produces a carboxylic acid group. It produces a carboxylic acid group. Okay, these are the some reactions of the these are some physiological reactions. These are the some reactions of physiological importance. So, for example, the D-ribose may make the nucleic acid and the metabolic intermediates. Ribulose may be important for the metabolic intermediates in the pentose phosphate pathway. Arabinose may be present in the plant gums and constitute the glycoproteins. And xylulose plant gums, proteoglycans and glycosamine glycans and constitute the glycoproteins. On the basis of the reactions, on the basis of the oxidation and reduction reactions, the sugar molecule can be divided into the reducing and the non-reducing sugars. Many oxidizing agents, including Tollens reagent, including silver ammonium complex and the cupric ions, are used to identify the presence of the reducing sugars. Reducing sugars are those sugars that contain a free aldehyde group and are capable of reducing metal ions in solution. This is the reaction of the reducing sugar. This is the reaction of the reducing sugars. Okay. Reduction of the aldehydes or ketone group of a carbohydrate is also of biological importance. The reduction of aldehyde group in glucose yield sorbitol, a sweetening agent. Another form of reducing carbohydrates are the oxy sugars. Here you can see this is the sugar we treated with the tolerant reagent. The reduction of the tolerance reagent containing the, say for example, silver molecule, or if we treat it with the copper molecule, it will be converted into the Cu2O. So because of this sugar, the reduction of these molecules can take place, and if we reduction of these molecules will take place. So on the basis of their reduction process, we can find out the presence of the reducing sugar. Say for example, in the failing solutions contain the copper molecule. The reduction of this molecule convert it into the Cu2O. So because of this, when this molecule Cu2O is formed, it is precipitated into the brick right brick brick red like substance. In the presence of this brick red precipitates, on the basis of these brick red precipitates, we can find out that in this solution reducing sugar is present or not. Esterification sugar can make the esters with the other molecules. It can they can make the esters with the other molecules. Esters are formed by a reaction of hydroxyl group with the acids. The most biological esters of the carbohydrates are the phosphate esters. You can the glucose 6 phosphate is the one of the major molecules which contain the which esterifies the phosphate molecule, phosphate group to the sugars. The next important reaction is the mutarotations. In the mutarotation, there are two major forms of the glucose in anomeric forms. One is the alpha D-glucose and second one is the beta D-glucose. These two anomeric forms can be interconverted into one another. Say, for example, the specific rotation of the alpha D-glucose is the 112 degree, and specific rotation for the beta D-glucose is the 18.7 degree. So both of these can be interconverted. Can be interconverted. This interconversion may be because of the addition of a water molecule or some other maybe because of the addition of water molecule. If you can see, or treatment with the other molecules. Say, for example, here if the water molecule is added, because of this addition of the water molecule, this hydroxyl group can change its position into the beta form. So this process is known as the beta. This process is known as the mutarotation. The next process is the enolization. Enolization is actually the process of in the process in which the glucose in which enolization is the process in which glucose when treated with the dilute alkalis, it can be converted into the several different forms. Uh, if we treat the glucose and let it for the several hours, a mixture of different sugars can also be obtained. Initially, we will see the fructose plus mannose. This is actually the conversion of one form of the monosaccharide into the another form of the monosaccharide. So for example, the glucose is being converted into the fructose. Because this conversion is through an intermediate molecule called the anidiol. So this process is known as the enolization. This process is also called the Robert-de Bruijn von Ickenstein transformation. Amino derivatives. The replacement of the hydroxyl group on a carbohydrate by an amino group results in an unusual class of the compounds, the amino sugars. Amino sugars that occur within nature are D2 amino glucose and D2 amino galactose molecules. Uh, One is known as the glucose amine and second one is known as the galactose amine. Glucose amines and its acetylated derivative, N-acetyl glucosamine and N-acetyl muramic acid, are found as structural compounds of the bacterial cell walls. Similarly, the N-acetyl glucosamine is a compound of chitin, a polymer found in the exoskeleton of the insects and the crustaceans, and it is also a major structural unit of the chondroitin sulfate, a polymeric compound of the cartilage. They are also present in the cartilage. The acidic amino sugar N-acetylmuramic acid, or you can call it the salicylic acid, is derived from the carbohydrate rhamnose and is a compound of the glycoprotein and the glycolipid. Glycoside formation is an important reaction of the monosaccharide in which by proceeding through this reaction the monosaccharides can make the chain of the molecules. I already tell you that we uh, divide the carbohydrate into three major molecules. One is the monosaccharide, which can now be further subdivided, and second one are the oligosaccharide, in which 2 to 20 subunits of the monosaccharides may be present, and the other one are the polysaccharides, which having the major starch and the glycogen molecules and can be uh, more than 20 subunits are present in these molecules. For making these all of these molecules, the linkage is very important. Among this linkage, the major linkage for polymerization of the carbohydrate is the glycoside linkage. O glycosides, when carbohydrates are reacted with the hydroxyl groups, under the anhydrous mildly acidic condition, a new linkage and glycosidic linkage is formed. If you can see, say for example, if these two are the molecules, uh, one, okay. Say for example, uh, this is one sugar molecule. Say for example, this is the glucose molecule. This is the glucose molecule, and second one is this one is the glucose molecule. When the hydroxyl group of this one and this one will react will react, a linkage between these two molecules through an oxygen atom will be established. So this linkage, this bond is known as the glycosidic bond. The hydroxyl group of another carbohydrate molecule can substitute for the alcohol in the reaction, linking two monosaccharides to make the disaccharide and the polysaccharide molecule. Okay, these are the diagrams related with the previous text. Okay, and glycosides. The NH group, the amine group of the DNA group of the amines can substitute for further hydroxyl group and react at the anomeric carbon center of the carbohydrate. This linkage is called an N-glycosidic linkage. The previous one was the O-glycosidic linkage. O-glycosidic linkage in which oxygen is being it was involved, but in this one, the NH of the amine group substitute the hydroxyl group react with the anomeric carbon center and make the N-glycosidic linkage. This type of bond is a paramount importance in the construction of nucleotides such as ATP and in the nucleic acid RNA and the DNA. Okay. Uh, Glycosides are widely distributed in nature. The glycons may be methanol, glycerol, and sterols, a phenol or base such as adenine. The glycosides that are important in the medicines because of their action on the heart, cardiac glycosides all contain steroids as the glycons. These include derivatives of the digitalis and strophanthus, such as the obians and an inhibitor of the sodium potassium ATPase of the cell membrane. Other glycosides include the antibiotics such as the streptomycin. This one is the O-glycosidic linkage. So, for example, this is one sugar molecule, this is the other sugar molecule, and between these two, the hydroxyl groups will interact and make the glycosidic linkage. The glycosidic linkage is of two types. One is the alpha type, and second one is the uh, beta type. These are the alpha O-glycosidic linkage, and there may be the beta O-glycosidic alpha and the beta O-glycosidic linkage. If if the alpha, if the first sugar contain, or we can say, if the sugar for which the aldehyde group or the keto group is being reacted uh, is in the alpha form, the linkage will be the alpha glycosidic linkage. If the sugar if the carbon if the functional group containing sugar, say for example the aldehyde containing sugar is in the beta form, say for example hydroxyl group was here. So for example, hydroxyl group was here. The linkage will be the linkage will be beta linkage. Say for example, this is the beta linkage. Here the alpha carbon was involved. The linkage is the alpha carbon. Here the alpha carbon is involved. The linkage is alpha carbon. Here is the beta carbon is involved. And here is the beta carbon. Similarly, in the N-glycosidic linkage, say for example, this is the amine. Amine is reacting with the glucose. Because of this reaction, this amine will will attach on the position of the hydroxyl group. So for example, this is an NHR. This is the here it is replacing the hydroxyl group. Similarly, this is so for example. Uh, this is the nitrogenous base compound. When it attach with the ribose sugar, it will make the N-glycosidic bond. Say for example, this is the adenine compound, adenine, and this is the pentose sugar, ribose. Say for example, then this is the ribose sugar. It will make when this adenine will attach with the ribose, it will make the glycosidic bond and glycosidic bond with polysaccharides. Now we are moving toward the polysaccharides. Hoping the next lecture will be on the polysaccharides. Up till now, this is enough for you. Thank you very much for watching this video. Thank you very much for looking this lecture. Hoping student, if you will have any problem, you will contact me. Thank you.